All right. This is from last week, but if you wanted to like screenshot it, that's fine. Um, but you might have already written this down. So that first one's atrial systole, then the ventricles are going to contract. And I put in here stroke volume. Not that stroke volume belongs there. Stroke volume is kind of its own thing outside of the cardiac cycle. But since we were talking about end diastolic volume and end systolic volume, then I thought I'd put it there. And then that's the last thing if you want to screenshot that. I didn't talk about isovolumetric relaxation, so you can skip that part. I was going to talk about it, but I didn't. So that's just kind of left over from last week. And I just want to finish out this chapter. And I didn't write this on the board, but I just kind of mentioned it, that cardiac output, it's your stroke volume, and then you take the multiply by a minute. All right, so you take stroke volume and then beats per minute, and then you get cardiac output. So that's another measure of cardiac output. I know there's like ejection fractions and things like that, but it's just another type. And this one, cardiac reserve, is just more FYI. That there's a big difference between like the minimum output and the maximum output. So it's, four times more. So this is new. So I'd like to talk about regulation. How do we regulate stroke volume? I was hinting at it last class, but I didn't write it down. So preload, contractility, afterload. Now we're talking about the ventricles here. So how much do the ventricles stretch before they contract? That's really the important part. Contractility is the forcefulness of the contraction. So there's some, for example, norepinephrine can increase that, right? So there's some mechanisms we have to increase contractility. Afterload is the one that's a little confusing, right? So if you think about a water balloon, like you're filling a water balloon up with water, and then you're going to take like the end of the water balloon and you're going to like squirt people with it, right? So preload is how much water you can get into the water balloon, right? Because the more it stretches, the more water you're getting into it, the more you're going to be able to like squirt people with the end of the water balloon. Contractility is like if I were to come up to the water balloon and like push on it, it's going to make the water come out even more. Afterload's a little bit different. So in order for the semilunar valves to open in the ventricles, there's pressure building up, right? And the ventricles are starting to like squeeze, like contract, but the semilunar valves aren't opening up yet. You want to get as much pressure behind that as possible before those semilunar valves finally pop open and all the blood surges out of the heart, right? So the more pressure you can get in the ventricle before the valves pop open, the more afterload you have, and the more stroke volume you're going to have. So that's what that means. Does that make sense? Does anyone have any questions about that one? The last thing on, let me see if I got a slide now. The last thing here is control of heart rate. So you've got nerves hooked up directly to your heart. You've got parasympathetic fibers and you've got sympathetic fibers. So if you remember, both sympathetic and parasympathetic are parts of your autonomic nervous system. One is sympathetic is fight or flight. Parasympathetic, they say rest and digest. So sympathetic stimulation is going to speed your heart. Parasympathetic stimulation is going to slow your heart. And your brain just does that based on information it receives. Right? So you're going to get information to your hypothalamus, and then you have a cardiovascular center here in your brainstem, and it's just wired up with your heart directly. So how does it know what's going on with the heart? Well, there's different receptors you have in your body. These receptors are, I would say, around the heart. The main places you can find those receptors are around the aorta and the carotids. So like right in this area here. So you've got 3 types of receptors. These 3 receptors are coming up again next chapter. Proprioceptors are monitoring like limb movement. So if I put my arms up, that's letting my brain know my arms are up. I need to increase my heart rate just a little bit because I now have to get blood up to my hands. So that's limb movement. Chemoreceptors are going to monitor things like pH, oxygen, carbon dioxide, and they're going to tell the brain to make the heart beat faster or slower. Baroreceptors are monitoring pressure. So if your pressure drops, again, that receptor is going to trigger the brainstem and make your heart beat faster. So these are just straight up. That's just straight up stimulation, like nervous stimulation. But there's also chemicals involved. I'm going to add in a slide. Yes. Yeah, if you got a question, just.
All right, I have a question before you get off the subject real quick.
Yeah.
So you said sympathetic is flight or flight, you said?
Yeah.
And then what you had said about the parasympathetic?
Rest and digest.
All right, thank you.
Okay. What are some chemicals that, let me let this person in. What are some chemicals that can control, what are some hormones that can control heart rate? You already know one of them. We've talked about it in last chapter. I'm going to wait for you guys.
Epinephrine and norepinephrine.
Good. Then you have minerals. These minerals, this is different from, so I'm not talking about like all of this. I don't have it on this one, right? I'm not talking about, because if you remember, sodium, potassium, calcium are used in generating the action potential. It's not like that. This is a different job of them. They're used here to regulate heart rate. So sodium and potassium can speed it up, and calcium can slow it down. I'm going to give you one more. I'm going to talk about it in a second after I let this person in. I don't know if I spelled that right. I'm going to say yes. Oh, apparently I didn't. Does it know? It does. So, AMP, atrial natriuretic peptide. What it does, it's produced by the heart. It's produced by the atria, as the name implies. And when there's too much pressure in the atria, it's going to want to lower pressure. So it's one of the few hormones that actually lowers blood pressure. So it's going to do everything the opposite of antidiuretic hormone, but it's also going to slow heart rate. because a faster heart rate is going to raise blood pressure. So here we want to slow it. So that's a hormonal or chemical control of heart rate. So there's a neurological control of heart rate, and there's a chemical control of heart rate. Any questions on that? Do you want to go over anything from heart physiology?
Are we going over anything for the test, or all this for the test?
This is for the test. So it's going to be blood and definitely heart physiology. I'm not sure about blood vessels, but I know this is going to be on it. So there's chemical control of heart rate. Oh, well, I'm sorry. Control of heart rate. There's a chemical and there's a neurological component to it. So there's chemical control and neurological control. Regulation of stroke volume. That's another question. And that's just kind of listing it out like this. What are the factors regulating stroke volume? Well, what is stroke volume? And then what are the factors regulating stroke volume? So stroke volume is the amount of blood ejected from the ventricles when they contract. You get it by taking end diastolic volume and subtracting end systolic volume. But that's essentially what it is. How much blood is getting pushed out of the heart every time the ventricles contract. What can control that? That's these are three factors that can control that, or that have an influence on it. Cardiac cycle is another question. So question number 3, and that's been I broke it up into 3 slides, but you can do it how you want. You're gonna have to. For this one, I want you to write it out. I listed stuff here, but I'm like, I explain it. So why are the ventricles in diastole? Well, because the whole heart is still relaxing, and the atrium are going to contract. So I want you to write it out, and then you want to contract you want to contract because you want to push the last bit of blood into the ventricles.
Are these slides going to be available to us or no?
I think they're on the videos.
And I have one question too. You had said that to write it out. So my question is, the slide you're on right now, you want us to write it out exactly how it is from like this slide, the next slide, and then the other 3 slides?
I'm sorry, I missed the question.
What I'm asking is, you on the slide right now for the 3rd question you had said. So what I'm asking is, you want us to write it out just like that, like on the test?
Well, yeah, for this one, write it out.
Okay.
For this one, I'm probably going to just put list.
Okay.
I mean, you should know what preload means, but the last one, these 2 slides, you're going to describe. It'll probably be described, right? But it's not like you have to write it out in sentences necessarily. You can just write about the receptors that are picking up what's going on with like the heart or blood pressure, and then all that goes up to the cardiovascular center in the brainstem, and then signals are sent right back to the heart again. So you've got receptors like around your carotids. Signal goes up to the brainstem. Brainstem comes back to the heart. You should know what the receptors are because that's going to come up also next chapter. This was 4. It's just like I wrote on the board on Wednesday. I have another PowerPoint with like a picture of this. Yeah. So, I mean, I already explained it, but I'm just going to do it again. So there's a cell, and I'm saying that everything in the cell is intracellular, everything out of the cell is extracellular, and then I'm telling you that these minerals, these electrolytes, they have places that they like to hang out. Potassium is intracellular, so it likes to hang out inside the cell. Sodium and calcium are both extracellular. So to start depolarization, these sodium channels are going to open. So see, all the sodium that was extracellular rushed into the cell. So that's going to cause depolarization. Now to keep it, some of the calcium is coming in, but some of the potassium is going out. So the potassium that's starting to leave is being mitigated by the calcium that's coming in. So that's holding it. And then lastly, we've switched places here, and you have sodium on the inside and you have potassium on the outside. That's causing repolarization. And then before they do the next heartbeat, they swap places and back to the original position again. And then it just goes back again. So original spot, sodium rushes in, that's going to cause depolarization. The cell's going to get shorter. Calcium's going to trickle in, that's going to hold that contraction. Potassium's going to leave, that's going to cause the cell to go back to its original uncontracted, relaxed position. And then they swap places before they begin the next one. So I'd like for you to be able to understand that. I don't know what question that was, but this one's the last heart one. This is the cardiac conduction system of the heart. And again, this one's just listing. And it's listed out here for you. The pacemaker is the SA node, the sinoatrial node. If it were to, for some reason, not to work, the atrial ventricular node right here can take over and act as a pacemaker. You're not going to be running any marathons, right? But it can work a bit. I mean, you can generate action potentials anywhere along here, right? But your SA node is, I mean, it's not correct. The SA node should lead it off. It's like the conductor. So it's starting off, right? Then it goes to the AV node, then the bundle of his, or the atrial AV bundle. Then it branches into these left and right branches when it's going down this septum here. And then as it curves around, we call these Purkinje fibers. And essentially, this is telling you that the atria contract and then the ventricles contract. So those are the questions from the heart. Those are all the questions from heart physiology. Any heart questions?
So will we have to know like all the parts of the heart or no?
For lab, you will. I won't
Okay.
really ask you that on the lecture test.
Gotcha. All right.
What would the test question sound like for this image?
This one?
Yeah.
List the parts of the cardiac conduction system.
Alrighty, thank you.
So listing them will be 1 through 5, right? I'm just making sure. Just before.
Yeah.
All right, okay.
So hopefully that'll be an easy one to get.
Does it matter if you want us to write out sinoatrial or SA node is fine?
You could put SA node, but in your head, you should know what it means. I mean, atrial, that word's pretty easy. Yeah, you could write it out. I mean, you can abbreviate it. All right, let me close this one. And let me open this one. So we're going to talk about the blood vessels. So now we're moving into the next chapter. So these are never very helpful for me, but they might be helpful for you. But just a reminder that we have oxygen-rich blood and we have oxygen-poor blood, or otherwise known as oxygenated and deoxygenated blood. At the level of the capillaries, you'll notice, like, look in the brain, right? Or let's look in the lungs. There's pulmonary capillaries, right? At some point, it's like purple, right? It's not oxygenated. It's not necessarily deoxygenated, because what's happening is that, in this case of the lungs, oxygen is coming into the cells. At the same time, carbon dioxide is going out of the cells. So it's not, the blood's not oxygenated, it's not deoxygenated. There is kind of like this middle ground. I think of it all like, I think of an artery like an interstate, and sometimes we call them arteries. Maybe not in Louisiana, but we call them arteries, right? So you could fit, they're really wide. They're like 4 lanes. You can fit a lot of cars on them. They're going very fast. There's not any place to stop except, you get off the exit. That's like an artery. That's how I envision an artery, right? And so you see lots of red blood cells there. And then as it gets smaller, it becomes an arterial, which I think of like Judge Perez or Paris Road. Like, you can go faster, but not as fast as you were on the interstate. That's like an arterial. So it's slowing. The idea of an arterial is to slow down the blood. So when you come over like the Green Bridge, you're still going like 70, right? But then you're going to hit that first light for like where the school is, and you're going to have to start slowing down. By the time you're at McDonald's and stuff, you're not going 70. You're going like 40 or 50 or something, right? And there's not 4 lanes anymore. There's just, on the interstate, there's 4 lanes, and an arterial, it's just 2 lanes. And then finally a capillary. That's where your house is. Your house is not literally on the interstate. Most of the time, your house is not on a major road, right? Like an arterial. No, it's going to be on a residential street, which is like a capillary. Sometimes these capillaries are so small that you could fit 1 blood vessel at a time on them, right? And then all these cells that I put here in light blue, that would be like the houses, right? So we have like the residential street, we have the interstitial fluid, which is like the grass in the driveway. That's interstitial fluid, and then the house is a cell. Therefore, the passengers in the car, in this analogy, would be what? If the car is the red blood cell, who are the passengers in the car? You should be able to picture me right now walking in between you up to your desk, trying to make eye contact. You're pretending like you're writing notes. So it'd be like the oxygen. It's like the passengers in the car would be like oxygen, right? So oxygen gets out of the car, walks across the front lawn, and goes into the house. The oxygen leaves the red blood cell, goes across the interstitial fluid, and enters the cell. At the same time, carbon dioxide is coming out of the house, out of the cell, and going into the blood. So I kind of turned these blood cells different colors. Does carbon dioxide literally ride on red blood cells? Only a little bit of it does, but I just colored coded it just so you can see. Because at the level of the capillary, there's oxygen going into the blood, and there's, oxygen leaving the blood, and then there's carbon dioxide coming into the blood. So that's why the color change, right? This is where oxygenated blood is going to become deoxygenated, and it goes in these little tiny venules, which are like capillaries, except now they're moving in a different direction. We're headed back toward the heart. And then all of those venules are going to converge into veins, which are a little wider, and all those veins are going to converge into the something. What do all the veins become? Go to the. Oh, look at you guys all with your cameras off. What do you think, Layla? What do all the veins converge into?
I have no idea.
What are the two vessels that go into the right atrium? Yeah, I'm just gonna say for you. That's not your fault.
Arteries.
All the arteries we're talking about on the return trip. So an artery going back to the heart, it's not going to be an artery, it's going to be a vein, and all the veins converge into the superior and inferior vena cava. So if I were to continue this vein and make it even larger, I would put vena cava. And what becomes the arteries? What part of the heart becomes like all of the arteries? So what's the first thing coming out of the left ventricle? Aorta. Very good. Nobody. Aorta becomes an artery. Artery becomes an arterial. Arterial becomes capillary. Capillaries are going to become venules once the blood becomes deoxygenated. Venules start to converge into larger veins, and in larger veins, they all come together to make larger vena cava, vena cava, superior or inferior, and then back to the heart. So that's kind of like a round trip. Here's all the arteries going out, right? So there's your aorta. We talked about this in lab. This is the one that starts with a B. Well, you can see them, brachiocephalic. And then that splits into your carotid and your subclavian. Here's your other carotid, and here's your other subclavian. And then you follow it down behind the heart, and then here's the aorta. You can call it like descending or thoracic. It's the aorta. And you see all these stuff branching off of it, right? So you see like renal arteries, gastric arteries, celiac arteries, celiac trunk, hepatic arteries. All these things are branching off of it. And then it splits down here into the iliac arteries, and then it goes down your leg. And you can kind of, the big ones are named after the bones, like so femoral, tibial, like that. Popliteal for the knees. So that's all the systemic. You notice the lungs aren't here, right? So this is a systemic flow, because the blood that's going to the lungs is going to be deoxygenated, so that's different. These are all the veins coming back. These are the arteries going out to the lungs, but look at the rest of these, right? So these are all coming back. Superior vena cava, inferior vena cava. All the veins, did I say arteries? All the veins are all going to converge into like a larger subclavian vein, and then that goes into the vena cava. So same setup, just this is deoxygenated blood, and this one's oxygenated blood. These arteries sit a lot deeper, and the veins sit a lot more superficial. But now let's get into let's start talking about each of these. Right, so let's look at the artery. Let's look at the artery. So the artery has 3 different coats, right? So a coat we call a tunic. So that's the names of it. Tunica interna, tunica media, tunica externa. So there's 3 coats. So an inner coat, a middle coat, and an outer coat. So let's start with the inner coat. So here we're talking about the lining of the blood vessel. This is where all the red blood cells are. So the lining of anything is going to be epithelium, epithelial cells, what line your stomach, what line your, I don't know, the inside of your mouth. They also line your blood vessels. So in the case of the blood vessels, we call these epithelial cells, we call them endothelium. So that's all it is, just epithelial cells. Epithelial cells are very often attached to a basement membrane. So there's your basement membrane. And now, what kind of looks like Swiss cheese, you have a thin layer of elastic here. So there's 3 layers to this tunica interna. Endothelium, basement membrane, which it's attached to, and then you have a layer of elastic underneath it. That's the first coat. So there's like 3 layers to this first coat. Tunica media. Let's look at that. So there's 2 layers to this. This muscle going around these blood vessels, right? Because these arteries can constrict and they can dilate. So blood vessels go through vasoconstriction and they go through vasodilation. Here, there's a thick layer of muscle and there's a thin layer of elastic. So here's like another layer of elastic. It's not always like that. This particular artery is further away from the heart. So it's right here. It's like a muscular artery, right? You don't want to constrict blood vessels that are next to your heart because that's going to impede the flow of blood coming out of your heart, right? You can't constrict an artery, or you don't want to constrict your carotids. That's like right there next to your heart. You're going to back up the ventricle and you're going to mess up the heart. So those are elastic arteries. So if you look at the tunica, so an elastic artery would have like a thin muscle layer right here and a really thick elastic layer. So it's like the opposite of what you're seeing here. This is a muscular artery, or we can call it a distributing artery because you can shut the arteries like in your arms and legs. You could shut them down and divert blood to like your core if you wanted. So not a lot of elastic, more muscle, but on the other side of that, you have elastic arteries closer to your heart. Those are elastic because you want them to stretch out. You want that, like, as the blood leaves the artery, you want it to like you want the artery to be able to stretch out a little bit, right? And then the artery, because it's elastic, it's going to snap back, and that's going to push the blood even further along. Right, so that's why we call them conducting, because they help. The elastic helps to like push the blood through your body. I hope that makes sense. We're not going to talk about arterioles, but there's one thing that arterioles do that you should know. They slow down the blood flow and they lower the pressure. It's the same thing, right? So you want to lower the pressure. You don't want the pressure that's in your artery. You don't want it in a capillary. If it's in a capillary, you're going to blow the capillary. Capillaries are really thin. Let's look at them. This is a capillary down here. It's essentially epithelial cells, and there's some basement membrane around it. No elastic, no muscle, no tunica externa layer, which is just connective tissue. You don't have any of that in a capillary. They break super easy. So you need to lower the pressure, and that's what arterioles do. So by the time, and arterioles have some ability to constrict, so they do constrict. For example, angiotensin 2. It constricts arterioles. So there's a lot of receptors on these for angiotensin 2. That's in particular what it constricts. But capillaries, no constriction. There's no muscles, no stretching, there's no elastic. They're going to break. So, so here we're at the capillaries, and there's a lot of, they're at the cell level, we call these things capillary beds, right? So in between all of these, like in these spaces in here, imagine a bunch of cells. Right? These are the residential streets in that analogy. Our body has the ability to, like I was saying, we could shut blood off to the arms and legs if we had to. Let this person in. One of the things that we would do is shut off those precapillary sphincters. So we shut these sphincters off, and we can send blood through this thoroughfare channel, put it right back into the vein, get it back towards the heart. So if we had to in an emergency, we can divert a lot of blood to our heart. And actually, a lot of our blood, if you look at like all of the blood where it is right now in your body, the majority of it is in veins. So we could send all that blood quickly up to your heart as well. So test question, no. I'm just kind of showing you that capillaries sit in a capillary bed. You have, sometimes you can directly get the blood back into venules, which is going back to the heart now, right? The venules are going to become veins, and the veins are going to become the vena cava. So these are capillaries. Again, endothelial cells, basement membrane. Well, there's 3 types of capillaries. So now I want you to start, that's all I have. Now I want you to start writing down the types of capillaries. Like this is stuff that I'll ask you test questions about. The first thing I would ask you a question about is the types of arteries. That's a small, easy question. Just think about it. Elastic arteries are going to be close to the heart because the blood is surging into them. They need to be elastic. You can't have a lot of muscle in those arteries because you don't want to constrict them. So further away from the heart, have muscle. Closer to the heart, more elastic. The 3 types of capillaries is another question. And I think we're going to take a break at this point after I talk about these types of capillaries. So, continuous. The only place where things can get in and out of continuous capillaries would be these little spaces in between the cells, which we call intercellular clefts. All right, so this is a cell right here. Here's another cell right here. And this is the little space between the cell. Space here as well. That's it. So not a whole lot of openings in this capillary compared to the other 2 you see. So this would be found like in the brain, for example. You're not trying to get things in and out, a lot of things in and out, right? Now we go to fenestrated, and fenestrated just means like windows, like little pores, right? And so the intracellular clefts, although it doesn't look like it here, they're a little bit larger. This little cleft is larger, and that cleft is larger. And then you have these fenestrations, these like pores. So now you can start to get more stuff in and out of the capillaries. So you might see these in endocrine glands when you want to put chemicals into the capillaries. So you can just put them through these holes. This last one's called the sinusoid. just take whatever you had for fenestrated and put really big. So really big fenestrations, really big intracellular clefts. Some of these are so big that it looks like you can get a blood cell to go through them. So this might be, you might find this type of capillary inside of a liver. Maybe I need to get whole blood cells out into the liver so the liver can eat them up. Or the spleen, or red bone marrow. Red bone marrow makes red blood cells. Where are you going to put these red blood cells after you make them? You need capillaries with like big holes so you can put the red blood cell inside. All right, so a sinusoid capillary, just really large fenestrations, really large extracellular clefts. Those are your 3 types of capillaries. Let's take a break. We'll come back like at 9:32. We'll come back like at 9:50, like for sure, or like a couple minutes before. And we'll start talking about some of the physiology of blood vessels, like how fluids get in and out of blood vessels, things like that. So
He said 9:50.
yeah. I'm going to turn my camera off, but I'm still around. In fact, maybe I should stop. I'm going to stop the recording, but we'll still be here. I'm going to stop the recording and then just don't let me forget to start it again